I wanted to welcome everyone to the webinar today. Thanks so much for joining. We will be discussing some really interesting topics via a fireside chat. Just to give you a little bit of context for those of you who haven't joined before, this is our Cyclica's Molecule to Medicine series, whereby every month we host either a lecture episode, which is the more academic kind of slide-driven learning quest, I would say, and you're tuning into the alternative, which is a fireside chat, which is more of a casual discussion about a thought leadership topic within the industry. As you can see, we cover a variety of topics within AI drug discovery. And today we are discussing what a neo-biotech is and kind of historically what a biotech has looked like, and moving forward what a biotech / neo-biotech will look like. So just to kind of set the stage slightly before I introduce the panelists, kind of an overview, historically, new biology versus new modalities, how there's a need for change within the current paradigm. We're going to talk a little bit about the dark proteome / low data targets, also the massive opportunity that awaits within repositioning, and what does the future look like? Where are we going to be in 10, 20 plus years? So we have two exciting panelists today. So we have Naheed, who is the president, CEO, and co-founder of Cyclica. And Naheed is also a co-founder and board member of Entheogenics, also the AAIH, and he sits on the Ontario Bioscience Innovation Organization board. He's also a member of the Life Science Advisory Group for Global Affairs Canada. So we look forward to hearing from what Naheed says. And without further ado, I'll pass it over to Naheed, who's going to introduce our second panelist, David DeGraaf.
Jen, thanks so much for that introduction and for welcoming everybody to, I think, episode 9 of Cyclica's Molecule to Medicine series. And for those of you who are new to this, you can visit the Cyclica website to watch or learn more from previous episodes of M2M. We also have it on Spotify as well as our YouTube channel, where this will go on as well. And the conversations that we generally have as part of M2M follow anything that happens kind of in the discovery of new medicines, from target ID all the way to preclinical development and then into the clinic as well. And as Jen mentioned, some of those are more specific on a topic. Some are more just conversational about trends that are happening. Today is one of the latter, a historical view, what's happening today and what the future looks like, aligned with how Jen introduced the topic. And I can't be more excited to talk to David DeGraaf, one of my close friends, a mentor, an advisor, somebody who I hold in the highest of esteem, who's taught me a lot about what not to do in the industry. And the inverse of what not to do is what to do. And Dave does a lot of that stuff of what to do right. And the conversations that we've had over the past probably 7 years, David, since we were introduced by mutual friends, the John Conways of the world, the late Frank Browns of the world, you've had a measurable impact on myself and therefore Cyclica. And I wanted to introduce you in this conversation to the listeners, either live and then thereafter, and kind of have them a virtual seat at the table. When we meet, it's always over a cocktail or a beverage, lunch or dinner, and we have open-ended conversations. So instead of having a pedantic back and forth Q&A, let's just imagine we have a group of dozens to hundreds of people sitting around the table, and it's just you and I having a fun conversation. How does that sound?
Yeah, that sounds fantastic. And I brought my coffee. There may be something poured in here to make it resemble a cocktail. So we're all
We have no old fashions here at the Cyclica office, though I'll be sipping my coffee thinking of that. So I'm not going to go through David's illustrious background. Some of it is on the screen today. But if I were to summarize David, and then I'll pass it to himself if he wants to introduce a little bit more, deep background in the life sciences and specifically in pharma and in biotech, having spent many years in big pharma companies taking that knowledge, that experience, that wisdom, and bringing it into the investment space in biotech, and then turning from an investor into an operator, having founded and built as CEO some prominent biotech companies, has some wins under his belt, and is now onto some very exciting things. Maybe we'll hear a little bit about that today as well. David, beyond that, anything that you'd like to say to introduce yourself?
No, it's just in general, I think that for most of us, we're very lucky to have the opportunity to help heal the world. And I think in the end, whatever we do there, it's really not about us. We're in a sacred industry. We get to make medicines for people who need it. And the better we do that, the faster we do that, the more we can serve the people who need it.
I love that. So let's start off with, and I can assure everybody there's no planned questions that have been sent to David or that I have planned. So we're going really off the cuff as though we just sat down, haven't seen each other for a couple months. So I'm going to pick up on that. That's your North Star. Was that always your North Star? How did you get into this industry? And talk us a little bit about the journey that you've embarked on personally.
Sure. It's so I'm a pharma brat. My father is a chemist by training and worked for Organon for 35 years. I lived in the town where Organon is. I couldn't wait to get out of there for a whole host of reasons. Wanted to study something different. And so I went to study biology, ended up doing a PhD in the US, postdoc in Israel, came back for my first faculty position, a junior faculty position at MIT, and saw biotech rise up around me. And I was convinced at that point in time that this was around technology, that technology was going to be the driver, that one way or another we needed to be smarter. I had a background in genetics and genomics, and I started to do the first more computational approaches, more classical bioinformatics. I made the move over to pharma to my own surprise, because I thought the opportunity would be in small biotech. I actually did a bunch of interviews there, and one very wise person, Maz Mansouri, said to me, David, you need to have a broader background in drug discovery and development. Why don't you first go and work for a pharma and then come back to me and we'll do a biotech together? I never got to do that with him. His son ended up being a board member many years later. But that's how I ended up at AstraZeneca. What I learned in pharma was to be very, again, very humble about what we can actually do. Like making medicines is an incredibly difficult thing. I always think of them as tiny machines, whether they're small molecules, antibodies, or gene therapy. They need to do a hell of a lot more than just fix an issue with an enzyme that needs to be inhibited or a gene that needs to be replaced. They need to get to a place in the body. They need to provide safety. You need to understand so much more about that molecule than just whether it works or not. So that became a big driver and really a fascination. Then over the last 12, 13 years, as I learned how to make drugs, I started to look at where I felt the largest unmet needs were. And I started to talk to patients with chronic diseases, mostly autoimmune diseases, but also a set of other diseases, started to see what that need was. And so it went from technology to really being driven by patients and patient needs. I keep in touch with patients, also patients from previous companies, just to see where their journey is taking them and to see what we can do to help them out, but also to keep on learning about what the real issues are for patients. So that's in brief, sort of what my motivation is and how things have changed. How about you, Naeem?
Oh, I was going to come up with a follow-on question to that. My story is a little bit more eclectic and less, I'd say, specific to entering this space with intention. But in summary, I do want to make this about you, but I will answer your question. My passion, since as long as I can remember, is in the biological foundations of behavior. Why do people make the decisions that they make, and how does biology impact that? And so in undergrad, I was steadfast in studying kind of brain disease, how the brain functions, and then copying that and getting into psychology and understanding how people's behavior in the face of new data and new information, how they interact with that data and information to make more informed, sometimes better decisions, sometimes worse decisions, and why does that exist? And ultimately, I was doing that to get into med school, like any good Indian student of my generation. There was law school, engineering, or med school as the three paths that our parents pushed us down. And I was studying for my MCAT, pulled the plug on that for some personal reasons. That then fast forward 10 years or 7 to 8 years led me to join my co-founder in creating Cyclica, which was my grandmother who suffered from lupus. For my entire relationship with her, was misdiagnosed or was misprescribed medications that counterreacted to her with her lupus meds. She slipped into a coma and passed away, which was then the reason why I left the med school track and got into business instead. And then fast forward, the opportunity with Cyclica to get into the discovery of better medicines, to minimize off-target profiles, to minimize adverse drug-drug interactions, which would be one of the initial thesis back 10 years ago. And that's what got me back into the space. I'm very much driven. If somebody were to tell me today, and this is being recorded, so I know that this will be live and into posterity, that Cyclica's technology would be instrumental in the discovery of a medicine that would reach a patient, and whether it be in a clinical setting, and that would treat or cure a disease, I'd say take it and go all day long. I'm not in this for the money. I did not start a company and kind of invest everything into it because there's a golden pot at the end. That golden pot to me is how can we do much more with technology and a new mindset to find medicines to treat and hopefully cure not just one disease, but hundreds of diseases. And I think that's where the power of new technology exists. And we can spend a little bit more time on that.
Right. Yeah, I think that's, there's so many interesting parallels, both culturally and in terms of what the drivers are, but there's nothing like understanding what a patient actually goes through to help drive you to want to do something about it. So that's, I think the first time somebody actually made that happen for me, it was Phil Vickers at, when I was working for M2M E09. Phil invited a patient with MS who had recently been diagnosed. This is a guy who worked on his car every day and said, how did you know that something was wrong? He said at a certain point, he was trying to turn a wrench and his hand just didn't work anymore. Just like that, stopped working. And just hearing his story was, first of all, very, very touching, but also started to make me realize that we're not just fixing a molecular defect, which is very easy to focus on, but these are patients' lives. And for a disease as complex as lupus, it gets to be really, really complex and really difficult to get that done. Anyways.
So coming back to you a little bit, you've been in the industry both as big pharma, investment, and now biotech across different modalities, different therapeutic areas. What is, if you could talk about the most important learning that you have had regarding the business of pharma, as well as the scientific discipline, and what have you learned about how best to navigate both? And who did you learn that from? Is there one individual that you learned something from? You mentioned Phil Vickers. I spent some time with him, back when he was at Shire. Always, really impressed with the way in which he thought about the world. And so are there more individuals like that that have kind of imprinted on you the right way of going about drug discovery?
Yeah, it's absolutely. And many people in many different ways. I don't know, I'm almost hesitating to mention names because I'm going to forget somebody who I realize is really important. My PhD and postdoc advisors, of course, taught me things that were pretty deep. Now, as a human being working in this industry, actually, Eric Lander, who brought me to Boston. Eric, people can have different opinions. As a thinker and a scientist, very few people where you'll feel so his sense of urgency to actually get things done. This was when we were sequencing the human genome. That sense of urgency, I wouldn't be able to get from anywhere else, because it's very easy in science to sort of hide behind the timelines. Oh, it's an experiment. We'll do another experiment and we'll figure it out. really pushing that become incredibly important. Obviously, my father and what he's taught me about his drive to go and try to get things done for 35 years to get a drug to market. And in industry, there are a whole host of people who've taught me. You have as well, right? So it's very hard to get to those small pieces. Here's the thing that I think most people don't understand about all of this. And that is that when you make a drug, it's really important to know why you fail. But because most of the time, 99% of the time, we're going to fail. We're not going to get that drug. To your point, like it would be great to have Cyclica drive that. There's a decent chance that that won't happen. But the problem becomes that we do so many experiments where we don't learn from failure. And that means already you need to set things up all the way from the beginning so you understand, so that you understand enough about your program and your drug that you can understand why things don't work. And so let me give just a practical example. I worked on a compound for a small biotech with some absolutely outstanding people that had some basic biological effects that were stunning for a set of rare genetic diseases. So in animal models of those rare genetic diseases, I don't love animal models, but for rare genetic diseases, it tends to be relatively predictive because it's the same lesion in the genome that you're looking at. This drug did amazing things. The thesis behind why it did it was completely wrong. Completely wrong. And that meant that when we moved it forward and we tried to optimize and do better, because that drug was never going to be a drug. It was a compound. It was never going to be a drug. We couldn't do it. We didn't know what we needed to fix. So having a deep understanding of mechanism becomes an incredibly important driver. And I've seen investors jump over that because you see great results in a disease. You don't completely understand why. And you're thinking, well, we'll just go and scale this. And I can give you countless examples where there was a great thesis, an unexpected result that gets moved into phase 2 or phase 3, and then complete abject failure. And that wastes patients' time and lives. It wastes resources. And we need to understand. And one of the things that always attracted me in working with you and the Cyclica team, just as an external advisor and friend, was this idea of actually starting to understand why compounds do something. Like having that direct link. So you have a place where you can start to fix your car. Right? If your car doesn't run, is it because there's no gasoline in there? Is it because you ran out of oil? Is it because there's an electrical defect? If you don't have that figured out, you can't open up the hood, how are you going to know how to fix your car? And we're in the same business.
So that leads me down a few questions. One is there's a lot of investment that is going into the biotech space, both in terms of new platforms for new biology, new platforms for new modalities, and we'll go down that in a moment. But I want to come back to your point on understanding mechanism.
Yeah.
A lot of investment has been put forward in the community and primarily by big pharma companies in phenotypic assays. Over the past 10 to 20 years, there's been ebbs and flows on the investment in phenotypic assays, but inherently coming out of a phenotypic assay, the question why can't necessarily be easily answered. And so, yet there's now a renewed push, I'd say over the past 2 to 3 years, at least my observation, I'd love to get your thoughts on this, where a number of folks are saying, can we bypass biophysical evaluation of a molecule to a target, understanding the why, and bypassing that to get to the what, which is in the phenotypic assay, and advancing without having to get necessarily a true understanding of mechanism of action. What do you say to that?
Well, so here's the thing. Here's the wonderful thing about being a small biotech. Now, you and I get to make decisions about which programs we work on and what we do. And so I would never work on a program where I don't understand the basic biophysical action that's underneath it, because I've seen too many of those programs go wrong. And I remember in my pharma days, we'd call that, when there was a phenotypic assay, you sort of understood at least some of the molecular inputs and outputs. We called it a gray box as opposed to a true black box. And so there are gradations of all of this as well, right? You understand a bit of the mechanism, you understand nothing. But in general, I feel that having that the ability to connect the dots all the way to what happens to the patient is incredibly important. So for me, that's a driver in my selection of programs and in my selection of new biology. And so that's the other thing is that we're chasing novelty constantly. But that's a stupid thing to do because novelty requires significant investments. All right. If you don't know anything, and I remember being so excited in my early days in pharma, saying, oh, we can do all of this target discovery. And whatever target we proposed would always lose out to a target that had 20 years of research and 5 labs working on it to go and validate it. We may have been right, but it needed that particular type of investment. Now, and that's actually a point where now with more complete data, still a very data poor science, but a more complete data and the ability to use machine learning and AI approaches, we can start to fill in some of those gaps for these dark targets, these low content targets, and actually start to fill in bits and pieces of that. And that becomes your ability to take one of those targets and put them in the other category.
Yeah, I love that answer. And it's something that I think informed the way in which we thought about the world for a long time. In fact, I'm dating the company a little bit in terms of, age, but also business. Our initial business, the one that you got to know 7 years ago, was on phenotypic screening target deconvolution because we felt that it was so important for various reasons that you've highlighted to identify the target of action for that specific biological readout or the disease or what have you in that phenotypic assay. And that without it, whether it be regulatory or investment or partnership, without that target of action, it would be very difficult to then advance that asset forward. I think we're seeing some thinking that is changing a little bit, a bit of a different appetite now, but still target of action, I think generally is very, very important. I'm going to switch gears for a second and get back to the question that I prefaced a little bit on investment in new biology and new modalities. You've been on both ends, biotech entrepreneur, building technology for modality and for biology and modality. You've also been an investor.
Yeah.
If you had a blank check right now in front of you and you could invest in one of those two areas, where would you invest and why?
Oh, God. Well, it obviously depends on the biology and the modality. But if I had set up a fund right now, so what you can see is if you look across new drug approvals for the last 3 or 4 years, there is barely any new biology in there. Most of it moves targets. If you're working on antibodies from monospecifics to bispecifics, if you go for small molecules, suddenly you can go after RAS or RAF, where this was always something that we've been beating our heads on, but we've known for 30 years that that's a great target. So if you actually look at where money has gone, and I think rightfully assuming, I think it was about 80% of new drug applications last year started their life in a small company, right? What we're investing in reality is we tend to invest in old, very proven biology, and we try to just do something new with it. And even there, the novelty is really incremental. It's break outside of the rule of 5. We're not talking about, we're going to make, we saw mRNA take a seat at the table. And I think over the last 20 years, we've seen very slowly how nucleic acids have made it. But we're still functionally dealing with nucleic acids, smallish molecules, and protein therapeutics. Everything else, there are lots of great, crazy ideas, whether it's now Rubius making red blood cells that make things, or whether it's gene therapy and cell therapy, either have made very limited or absolutely no impact, right? And so what I'm seeing is, even when I'm pitching right now, I'm actually pitching an old target. We're going after a cytokine with Rebirth Therapeutics. That's my new company that actually has significant clinical evidence. I think there the issue has been people haven't really dealt very well with the fact that administration can be very toxic, and we hope to be differentiated there. So let me say, no, I think we've got something novel to contribute, but we're not taking biology risk. And if you then look at the other side and you say, how many of these completely novel targets actually get financed and make an impact, your chances of getting to the market are much lower. You can see what has happened with TIGITs in IO. If you start to think through, now in general, what has happened with IO targets, tons of work, tons of biology, absolutely zero success in adding to the list of biologically validated targets. I think a little bit of that means that what we've seen increasingly is that people like to go after clinical precedent. That means that they sort of change incrementally the modalities to try to get them a little bit more refined, a little bit better. And the idea to really go after new biology has been really, really hard.
So that sounds like relying on decades of experience and investment to uncover biology, trust that biology, and bring in a new approach to drug that biology.
Yep.
Do you see a paradigm shifting to radically adjust to uncover new biology faster so we don't have to wait 30 years?
Yeah, I mean, that's what neo-biotechs, at least that's part of what neo-biotechs, exactly what we need to do. And this is the promise of AI. So we were chatting a little bit before we had this fireside chat about, I found a new scheduling agent that's an AI scheduling agent that sets up meetings, right? I could not imagine, and most people who interact with it have no idea. It goes by email, human language is there. And now we've learned, and I haven't seen this in the press yet, but people are saying that ChatGPT has passed the medical boards in the US. Now, if that's true, you can already see that the idea of sort of bringing knowledge together and then knowing how to extract that will be insanely powerful. Really hasn't happened for biology at all, right?
And so there are some remarkable companies that are in the space of identifying plausible new targets for disease, moving beyond the high data targets and unlocking that protein universe. And in the numbers, at least that we've identified and rely on at Cyclica, is that about 7% of the proteome is of this high quality, high data target space, which is where a vast majority of the continued investment in new modality, new technologies for modality are going after kind of the next best in class for that target space. But then if you zoom out from that like lake of congested targets where everybody's trying to go after it, and you zoom out to this unexplored ocean of the low data protein space, we're seeing that, a lot of the targets there are really interesting from a genetics and genomics perspective, but are structurally underrepresented, don't have necessarily clear binding pockets, don't have co-crystal structures or a lot of ligand and chemical information around it. And as a result, are oftentimes incompatible to or recalcitrant to the existing technologies, whether it be DEL systems or HTS or ASMS or even AI-based drug discovery. And so from a biology standpoint, there's been a lot of investment in genetics and genomics, these really interesting targets, but no modality technology to really go after them. And so again, from an investment standpoint, would you rather find the next best in class target for a high data target? Or what do you think of the opportunity of creating like that first in class, probing those targets, validating the biology, or going after something new?
Yeah, especially if you can do that in highly parallel ways, it's all around use of capital, right? But there's one incredibly important thing that we've done as we've moved drug discovery in particular from large pharma to small biotech. So the way investors work is they like to take isolated risks. And this gets again to what we said about mechanism. If there are too many flips, it doesn't work. So going to somebody and say, I've got new biology to prove, and if that new biology is right, I'm going to need a new modality to address it, is too risky, because now if things go wrong, they don't know whether it was their investment in the new modality or whether it was their investment in the new biology, and they can't recoup either one of them because you don't have the right target. So that's why new modalities almost always get paired with well-known biology, And new biology almost always gets paired with very, been there, done that modalities. That's the way you can put the story together. So I think the search becomes, where can we find traditional small molecules or things that are a little bit outside of the rule of five that address the dark proteome, the low content targets. Or maybe, once in a while it will be an antibody or an antisense, but it should be something that's relatively straightforward to do. And that becomes a real challenge as you go and build these companies, because there are going to be places where you're going to have great, well-validated biology and no way to address it. So there's a target that I worked on many, many years ago at a large pharma company. Let me not identify it too much. It was one of the IAP proteins, a protein called survivin. Great data that this is a fantastic cancer target. I don't know what has happened in the last 15 years since we looked at it, but we could not find out how to drug survivin. IAPs in general, they're pretty much sticks. Really, really hard to go and think about it. If that's solvable, you're off to the races. And that may not be completely dark, that may be somewhere in the middle, but these are the types of problems that I think you can start to move forward. And it's really, it's always about just having a wedge to start to pry that biology apart.
Yeah. And continuing on the biology, your background obviously is very diverse, but, academically, systems biology is an area that, that's where you grew up. And so you think about the world of protein structure determination right now, it's a very hot space. DeepMind and what they've done with AlphaFold 2 and, the other technologies in protein structure determination. From your perspective, in the seat that you have looking at biology and targets and then trying to find new ways to drug them, how do you think about the expansion of our biology understanding through the advent and availability of things like AlphaFold 2?
Oh, yeah. So that's what we need to do. So all of these layers of data need to be used to go and validate targets as early on as possible. And what I mean by validating here is validating them as a target, as a potential way to address this. Because I remember the discussion around validation I had when I was a couple of years into pharma, and one of the sales guys says, when a target is validated, when it makes $1 billion in the marketplace. That was his idea of validation. So what I'm talking about here is that you can link that target to disease biology in an incredible way, right? That particular gap. And yes, structural data are incredibly important there. And again, when you look at proteins, when I just called something a stick, proteins breathe, they move, right? They do things. And your ability to understand where that movement is and how you can fix positions so that you can, push biology one direction or another, obviously, is incredibly important.
Yeah, I like that a lot, and so much more to unpack. I wish I was deeper to be able to go deeper on those questions, but here's my cue to pivot to the next conversation: is the shift in what is a biotech company, from what was to what is to what should be going forward. This really well, and you made reference to that 80% number, that a vast majority of the discovery of new targets and the discovery of the molecules that oftentimes get to the clinic and become medicines, that innovation does not happen within the four walls of a pharma company. IQVIA had published some data back in 2018, 2019 timeframe, where they showed in the year 2000, about 50% of that work happened outside of big pharma. In 2018, it was 75%. I'm a strong believer, and I think there's anecdotal evidence that it's north of 90%. Yeah. And you're seeing biotech companies take it so far and then hand it over to a big pharma company who have the infrastructure to further develop and get it to the patient. I think Nimbus Therapeutics and what they did with their TYK2 asset with Takeda is a great example. They know their lane. They did it exceptionally well and monetize, and now another group is taking that on. And so as you think about the evolution of what is a biotech company today and where it's going, it used to be single target, find that one molecule, get it to a point, partner. And the investment was know your biology, know that target, find that molecule. Where are we today? And does that have to be the paradigm going forward?
Nope. No, actually, and I think that's again where you guys have made me think about, where Cyclica has made me think about the world quite differently than how I used to think about it. So we tend to think about how we move a product forward and about success and what are the steps where we have some validation that things will go and move forward. But in reality, we've got a lot of failure. So if we look at the small molecule space, about 40% of programs will fail in phase 1 because of explicit tox. Right? It passed through animals and it got there, and there's something wrong with these molecules. Then another, I'll keep it in the same sets of percentages. Another half of those will drop off in phase 2 because the efficacy cannot be achieved. And very often, again, that's related to therapeutic window. Sometimes it's related to other factors, but therapeutic windows is a big part of the problem that's there. And then even from there to phase 3, you see drop-offs that have to do with basic biology. So what I'm taking out of this is that sometimes you stop programs because there's no longer a commercial fit, there's just change in strategy, all those have impacts as well. But there's a whole host of biology that only hits us when we're in humans. And actually, if you could make even a small dent there by altering how you select molecules early on, you would have so much more success than what we have right now. And so for me, the big thing is how do we think about therapeutic windows in humans? And let me tell you, I'm not the only one. The FDA just said that they will start to accept submissions without animal safety data. Now, what those will look like, nobody knows. But is that an opportunity to start to say, can we put together a better way of assessing therapeutic window and the potential for risk, biological risk in humans than what we have right now? And that's absolutely the answer is we all think we can get there. And that, I think, becomes the opportunity because that will redesign our process completely. So let me give you a couple of other reasons why things fail that we can address early on. So developability of a molecule. And for small molecules, it's less of an issue right now because our chemistry has become so amazing. But for most other modalities, or for many other modalities, you'll end up hitting the clinic with your little batch that you made. Then you need to scale. You just can't do it. You can't do it fast enough. You can't do it well enough. Again, if you could address that risk early on, you could either scale up if you decide the opportunity is big enough, or you could just leave it out. So the first time this struck me was when I took over a global effort in protein therapeutics, when I was global head of protein therapeutics for Boehringer Ingelheim. And we realized that the paradigm that the company has set for how to move molecules forward was completely based on small molecules. And it was just a different rationale for large molecules. You could do different things. And I remember learning from our head of protein engineering, Sanjay Singh. At a certain point, I was in the lab and we were making the drug that's now Skyrizi. So it's an antibody against the IL-23. And I saw Sanjay was pipetting and then taking the little tubes, the Eppendorf tubes, and putting them on a high shelf. I said, Sanjay, what are you doing? He said, yeah, these are new clones. I'm just sort of informally testing stability. So in 2 weeks, I'll see how much of that is degraded. Just a very simple thing that we never want to do or need to do with small molecules, and we need to redesign. We, as an industry, have gotten locked into a process. So if you go and talk to VCs about what's necessary, what are the things that you'll hear? Well, I'd really like to see an efficacy model in an animal before I move my molecule forward. And this is a soapbox for me. I can tell you that I've not seen a single disease outside of some rare genetic diseases where the efficacy model predicts what will happen in humans. So where you can take classes of drugs that work in humans and that will work in the animal model, take classes of drugs that don't work in humans and they don't work in the animal model. Just doesn't exist. Doesn't exist for autoimmune diseases. Sure as hell doesn't exist for cancer. We all know that, right?
Brain disease, mental health.
Why are we spending money and time on that when we could actually go into much deeper biology and understand what likely is going to happen in humans? So there's something. When we talk about safety, what do? We dose extremely high in in animal models, and we hope that one way or another that tells us something about what happens in humans. We've got organs on a chip. We've got the ability to understand the difference in the specificity of the molecule in an animal versus a human because of technologies like the Cyclica technology. And we're not using those opportunities to, again, decrease that attrition that we see later on. So I'll get off the soapbox now because otherwise I'll be talking until the end of this meeting. But that's a real key. It's like we have the chance to think through and redesign this process and optimize it so that we do it faster, better, cheaper, and with more honesty towards patients who we're using to advance our research.
David, thank you for that. That's highly informative. I want to come back to something you said halfway through, which is new regulation that was approved by FDA on getting away from animal models, and the activity that the Alliance for Artificial Intelligence in Healthcare, the AIH, for which Cyclica is one of the co-founders, and now it's a member-driven organization of over 50 companies and institutions globally, big pharma companies, hyper-innovative biotech, academic institutions, and everyone else that is kind of around the AI and healthcare space. One of the big activities that our regulatory working group, led by a number of individuals, Paul Howard, Senior Policy Advisor at Amicus Therapeutics, and Meredith Brown-Tuttle, former VP of Regulatory Affairs at Recursion and now Chief Regulatory Strategist at the Regulatorium. They're leading an effort on thought leadership on digitizing the IND, because now what has happened is that there's this open space with this new regulation that was unanimously approved by both the Senate and the House in the US towards the end of 2022, which is now, how does this actually work? What does that look like? And we know the big why is that companies are using a variety of AI, ML, informed models to help make preclinical drug discovery development more streamlined, or a more streamlined process. And we are seeing that it's more likely to predict downstream clinical success. And the hope with the AIH's digitizing IND is that we can secure an FDA IND in a fraction of the traditional timeframes. And so we at the AIH are going to continue to put out thought leadership on how we can replace existing regulatory gold standard tools that can be extremely time consuming. Also in the process of that, save a lot of animal lives that unnecessarily, are victimized when the actual outcomes don't translate into human clinical success. And so I just wanted to make that comment, and a shameless plug for the AIH and for any active participant in this conversation today. If you're interested in learning more about the AIH, feel free to reach out, visit the AAIH.org, and we're always looking to continue to bring on new members to the organization. So David, please go ahead.
No. So continuing from there on, one of the things you need to do is you need to actually know why things have failed and how they have failed, because that will inform so much about your molecule. So I've heard some numbers, and again, I don't have these firmly from a citation, so I take them with a grain of salt. But there have been clinical trials on well over 10,000 small molecule compounds that were designed to hit at least 5,000 different targets. So a good number of those went through phase one. Let's say it's about 40%. And so now we have molecules that can intervene or interfere with at least 2,000 different targets in the human genome. And that means that if we would want to get started after phase one with compounds that are more safe, we actually have a whole lot of biology that we can explore, because we don't have 2,000 targets that are on the market. There's a huge gap there where we know we've got molecules that at least were designed to do something, whether they do it or not. That's a place where Cyclica can step in and then say, can we use these to go and perturb human biology in some meaningful way to affect the disease endpoint? And that opportunity to drop in the middle is really, again, enabled by deep knowledge of prior compounds, and then by the ability to take a new compound and say, can I find out what else this might do in the context of human biology?
Yeah, and the term that's used in the industry, and I think it's when used, it could denigrate the company that uses it, which is drug repurposing. And
Right.
for whatever reason, repurposing or repositioning assets, the term I like to use is reimagining new use cases for existing molecules. That entire concept is frequently deployed, but when it is talked about, especially with VCs, oh, we're doing drug repurposing, their mind blows like, we don't want any of that. And so it's interesting to see what's actually in practice and what other folks believe is the future.
Yeah.
And it comes back to, defining what is drug repurposing. And there's some incredible work that's being done by our drug discovery team, led by Mike Palovich, our chief science officer, head of drug discovery, and on his team, Julie Owen, who leads the repurposing franchise, on identifying like a drug-centric repositioning opportunity. And if I were to summarize, there's kind of three ways that we think about drug repositioning. One is a target-centric repositioning approach, where you have a molecule for a target molecule, one for target X, and now finding a way that, understanding target X is implicated in disease Y, but it's also implicated in disease Z. And can we now take that molecule and extend its indication from disease Y into disease Z on that same target? That's interesting. And there's a lot of effort into drug repurposing in that way. There is disease-centric drug repositioning, where you have a molecule that is effective for a specific disease, been shown in phase 1, phase 2, phase 3, maybe it's on the market. And now can you extend that molecule into a similar disease? So from one cancer to another cancer. Interesting approach, and a lot of repositioning happens there. I'd say the two of those, target-centric and disease-centric, represent about 90% of the effort in drug repurposing. What is less frequently used, and what we are strong believers at, is drug-centric repositioning, the idea of polypharmacology of a molecule, that a molecule will interact with more than its intended target when placed into a complex biological system. Some of those targets may be linked to toxicity, and you want to avoid those. So those are interesting questions to understand out of the gate. But then also there are targets that are unknown, unanticipated, that are linked to diseases that were not obvious to begin with. And so can you take a molecule for disease Y and for target Y and extend it for target Z and identify a new target for a new disease? And I think that's super interesting. And we have a lot of examples for that. But that means we don't need to renew or go de novo. You can take a lot of learning from assets that are in the clinic or where there's information that is available, and then now start evaluating for each one, which other possible opportunities that were non-obvious to begin with can we extend the indication for that molecule. And I'm really excited about that. I don't want to go into too much detail because this is more about, not Cyclica and more of a general conversation, but repositioning is, I think, the future of our industry.
No, well, let me just harp on that a little bit more, because I think you're absolutely right. So the question is always, do we have a tool to go and ask a question about whether we can help? And what we just said is we have tools, and understanding where we can use those tools. Cyclica has a very unique angle on, right? Because you can look at other targets. The first thing that I'd say is every single small molecule, by definition, will have polypharmacology. And it's not only driven by what people think of, which is, does it bind something else? It's also driven by avidity, how much of the target is actually there. It's also driven by how the molecule distributes throughout the body, right? If it never gets to the brain, it doesn't matter if there's a target in the brain. So there's some really basic pieces that make this a really interesting area to explore. And it requires what you do combined with some really great, smart, and very straightforward assays so that you can start to then think about repositioning those molecules. There was an ALS drug that failed for Biogen. And a couple of years ago, somebody was looking through the clinical data and they'd done bloods. And so this person saw that the compound was exquisitely, specifically affecting eosinophils. Actually, eosinophils disappeared in these patients. And eosinophils play a big role in asthma. So the compound was acquired by a number of investors. They put $350 million in for a small molecule in asthma. Companies called Aratea as an example of what you can do if you find something that's that interesting. So the question becomes, how was that done? It was essentially just complete luck. Somebody looked through the clinical data and made that observation. If you could do that systematically, how much more could you find? So that's one. The second thing is, if the compound does what it's supposed to do, but it's crappy, maybe you can make a better version of that molecule that does exactly what it's supposed to do. So now you have a fast follower approach to your own initial drug. And then the last thing is that we're talking about drugs that have already been used in patients and sometimes are on the market. Can you look at broad populations and EHRs and just do look at adjusted risk for people that have the disease that you're interested in, who happen to be on that drug or on that class of drugs anyways. So those are all ways in which we can validate and move forward. And we're not talking about something that nobody in biotech is doing. That's a neo-biotech right there.
Love it. So I recognize that we're 8 minutes till the top of the hour, and there are a couple of questions that came through the chat. So I will get to both those questions momentarily, just to tie the knot on the repositioning, repurposing angle. I remember a conversation at one of the first AI and drug discovery conferences back in 2017, 2018 timeframe, where the SVP of drug discovery at a big pharma company on stage said that this entire world of going after targets with new molecule discovery as the basis of drug discovery is leaving a lot on the table, a lot of value on the table. And his comment that stuck with me, and again, I'm paraphrasing what is now a 5-year-old quote in my mind, is that there is a drug that has already reached patients that for whatever reason failed, has now been shelved. It could be strategic, it could be efficacy for that initial indication, but there's enough work that's already been done where there should be a drug for every disease already out there. We just now need to find the right home for all that work. And so how can you bypass the lengthy, onerous, very costly, lengthy, time-consuming effort of de novo drug discovery to supercharge it through a reimagining new use cases of existing drugs, and then continuing that with a fast follower strategy if required, or et cetera, et cetera. I think that's the new paradigm. And hopefully what science shows, also the investors come through and say, yeah, we can get behind that, provided that a bunch of the checks boxes can be addressed, including how do you get over IP? How do you ensure the commercialization strategy? So there's still a lot to be answered, but from scientific first principles, it's a really interesting opportunity. I want to get to Paul Howard's question, and his question is for David. What role do sensors and AI have in helping to strengthen our understanding of the genome to phenome? And what role can that play in accelerating drug discovery in areas where patient heterogeneity is really a barrier to development? Big question. But he says, in other words, what looks like a single disease with a clinical phenotype turns out to be a cluster of different disorders?
Paul, I think it's a great question, and there's no easy answer. We define diseases by shared phenomena, and we already found out in cancer that we're usually wrong. That we group cancers mostly by the organ in which they occur. That turns out not to be. It's not the only, and may not even be the most important reason why those cancers occur. Right? They may have to do with just primary mutations. What went wrong? They may have to do with interactions with the immune system. So this is it's a puzzle where we both start with the pieces on the outside, which is really what we talked about. How can we build a framework for drug discovery? And also we're going to need to look at some of the pictures in the middle of the puzzle and start to put those pieces together. And we need to do both. So let me tell you how I get around it as an investor, because I don't have an easy answer for this. So as an investor or as an operator, because I make very similar choices because I want the investors to invest in my company, I select diseases with unambiguous, very clear readouts. And so my big driver is, do I understand that there's an endpoint that's shared by these patients that will be affected in disease and is registerable? And in general, that means that I focus on hematological conditions or on dermatological conditions, because in those cases, it's unambiguous. I've been working on a muscle disease just because of how severe the disease is and the shared underlying mechanism, a disease called inclusion body myositis. And the issue there is that we have horrible tools to measure progress for these patients. So it's not even or only patient heterogeneity, which is absolutely Paul's issue, but it's also just, if you understand your endpoint and you can unambiguously measure it, your program is going to be far more successful. And for those for those indications and those patients where that's not the case, we're going to need to really define what true benefit is and get that as an endpoint and start to point towards that, not necessarily the disease itself. True for neurological disease, true for muscle disease, true for things like diabetes and obesity, where you constantly have to take into account placebo effects and the heterogeneity of disease.
Love that answer. And Paul, thanks for asking that question. It is uncanny that I mentioned Paul Howard's name as a co-chair of the regulatory working group at the AIH, and then looked into the chat and there's a question from him. I did not know you were on this call when I mentioned your name. So hopefully I described the work correctly on digitizing IND. David, to finish off with the last 2 minutes, we have a lot of students that listen to the M2M session, people that are looking for inspiration, looking for guidance, perspective. You've said something today multiple times that I think is just brilliant, especially as a kind of an AI native drug discovery company at Cyclica, which is, you need to learn from failures. And that to me is very important because you can't update a, Bayes model is a machine learning model. You can't fully be informed if you're not looking at negative data as well.
Absolutely.
And so share with us in the last 2 minutes, one thing that you would encourage the future generation to continue to invest in based on an immense amount of learning and maybe some failure in your career.
Oh yeah, absolutely. So I've failed a lot more than I've succeeded. And I'm still very lucky to have been involved with now 3 marketed products with a bunch of things that have worked out. So the first thing I've learned over time is that it's better to be really good at 2 things than to be absolutely great at 1 thing. And what I mean is that as you build your career, stand on the interfaces. That's where true innovation happens. Just like Cyclica is built on the interface between machine learning and deep, intense biology, position yourself there as well. And I think a lot of the mistakes that I see people make is they are immense experts at a very small piece of what goes on. The second thing is, in terms of just learning and applying that more broadly, is I would say, if you don't care, it's going to fail. So all of the failures that I've seen, ultimately, it was either a scientist, an investor, a company that did not care about what was being done. So surround yourself with people who are aligned, who care about what you care about, and that can be caring about each other, but it should be sure as hell caring about what the company does. And then the last thing that I'd say, and it's sort of related to this, is if you care, it means you're going to be doing a whole host of things that you weren't trained for. And don't walk away from those. Now, pick them up, learn how to do them. And especially in small companies with small teams, you're going to need to do some things that you're all incredibly uncomfortable with because there's nobody else who's going to do it. So care deeply.
Excellent. And I think that's a fantastic way to end what has been a fun, insightful, illuminating conversation for myself, and I hope for everybody else online. David, thank you for sharing your time, your knowledge, your experience, your wisdom with a bunch of folks. M2M is something that we care about deeply, both from an educational standpoint, as well as from an educational inspiration, as well as boots on the ground, what needs to be done to move the needle in our industry. And there's nobody better that I can imagine talking to today than yourself. So thank you so much.
Well, and thank you so much. And for anybody else out there, at some point, I'll tell you about Reverb Therapeutics. We're still stealth, but very soon I'll tell the story. So follow me on LinkedIn. Okay.
Wonderful.
Thank you, so
David.
much.
Thanks to the Cyclica marketing
team,
team,
great discussion,
for putting this on. You're welcome.
Okay.
Cheers.
Yes. Thank you. Bye, guys.